Hello friends, I am Sagar Sengupta. I am the Director of National Institute of Biomedical Genomics, which is in Kalyani in West Bengal. I welcome you for today's whatever I have to say regarding our institute. Our institute was made in 2009 and we are now entering into the 14th year. We are the premier genomics institute in India. And we actually not only work on genomes, but we also try to understand the functioning of the genomes. So if I can give you some ideas how many genomes we have sequenced. We have sequenced close to 2,65,000 human genomes we have sequenced, which is around 8 lakh gigabytes of the data. It's a huge number beyond comprehension, and that is what our institute is mostly known for. We have contributed hugely to the India's effort in fighting the coronavirus, and that has helped India to become self-dependent, Atmanirbhar Bharat, to fight this deadly pandemic. In NIBMG, we have PhD and IPhD courses, and we have around 70 people who are staying in the institute and taking their dreams forward. We work on multiple aspects. For example, we work on the functionalization of the genome, as I said. We work on host pathogen interactions. We work on complex disorders, and of course, we have a very a good effort on statistical ways by which we can analyze the genome. So friends, come over to NIBMG, visit us, and if you are keen, join us in the onward journey. Thank you.
So in the National Institute of Biomedical Genomics, we have always focused on using and deploying cutting-edge genomic technologies for undertaking our research on genomics of human health and disease. In the process, right from our establishment, we have engaged and established heavily and developed expertise on massively parallel DNA sequencing, or known as next generation sequencing. We started off with various generations of these next generation sequencing platforms, and are we are at the global standard. Whenever a new method comes which can answer more questions, we usually take a deep look at it and invest. So this led to the formation of the National Genomics Core at NIBMG, which now not only caters to all the large and small projects on genomics in NIBMG itself, but also on other requirements of cutting-edge technologies of genomics for the academia as well as for the industry. In the process, what we are trying to do is to come up with a substantial promotion of genomics research in the country. Our center is the highest throughput genomics center in India, and we are fueling a lot of large throughput projects like the International Cancer Genome Project and subsequently many other projects. Recently, during the pandemic, our facilities are being heavily used for sequencing SARS-CoV-2 viral genomes in large numbers, and we have been monitoring the pandemic using genomic methods using these technologies. To add to all, we also have introduced high-end automation so that we can increase our throughput and at the same time have reproducible generation of data which will add to the quality of data and the quality of inferences that can be made from such genomics data.
Welcome to the NIBMG data center. So as I have spoken already, that in the lab we generate a lot of genomic data in those genome sequences. The same data flows back into the data center through cables, and once we get this data, we store inside these data centers.
The data that we generate in the lab that flows back to the data center, and then we at a remote location in the data analysis center, our students sit here and they use standard Linux terminal to analyze this data. So I'll ask show you how they are analyzing genomic data. So right now he's looking at the DNA sequence and trying to look at how the quality looks like.
Welcome to the Human Microbiome Research Facility of NIBMG. This facility is developed for doing human microbiome research and the functionalization of the genomic findings from the sequence results.
Welcome to NIBMG Proteomics Core Facility. The NIBMG Proteomics Facility provides a full proteomic infrastructure for the identification, characterization, and quantification of proteins and proteomes. The Proteomics Facility provides state-of-the-art mass spectrometry analysis of proteins for both basic and clinical research. Our Proteomics Facility is equipped with a thermoscientific Ultimate 3000 nanorapid separation liquid chromatography system online coupled to thermoscientific Q-extractive plus Orbitrap mass spectrometer. The facility provides unique opportunities for researchers at NIBMG and beyond to perform quantitative analysis of proteins in complex biological samples using both global as well as targeted proteomic approaches.